Welcome to one of the most important units in your A-level biology course, Control and Coordination. To survive, organisms must constantly monitor their internal and external environments and respond appropriately to changes. In this A2 unit, we leave broad physiological concepts behind and zoom right into the exact cellular and molecular mechanisms of communication. You will master the electrical impulses of the nervous system, the sliding filaments of muscle contraction, and the chemical messengers used by both mammals and plants.
We begin our detailed study with the fastest communication network in your body, the mammalian nervous system. You need to know exactly how sensory, relay, and motor neurons are uniquely adapted to transmit signals at high speed across the body. We will look at how the axon is wrapped in a myelin sheath made of Schwann cells. This isn't just insulation, it prevents ion leakage and forces the electrical signal to jump between the nodes of Ranvier, dramatically speeding up transmission through saltatory conduction.
But how does a signal actually travel along these neurons? What exactly is a nerve impulse? It is not a physical object moving down a tube. It is a self-regenerating wave of electrical depolarization. You will learn how the sodium-potassium pump maintains a resting potential of -70 millivolts, and how voltage-gated channels suddenly open to flood the cell with sodium ions. This creates the explosive spike of an action potential, which propagates in one direction down the axon, followed by rapid repolarization.
When that electrical wave reaches the end of the neuron, it hits a physical gap called a synapse. Here, the electrical signal must convert into a chemical one. You will map the exact chronological sequence, from calcium ions diffusing into the presynaptic knob to vesicles releasing the neurotransmitter acetylcholine. We will also examine the ultimate destination for many of these motor impulses, striated muscle. You will learn how these signals trigger the sliding filament model, detailing the specific roles of calcium, troponin, and ATP to translate cellular communication into mechanical movement.
Alongside the nervous system, mammals use the endocrine system for slower, widespread, and longer-lasting coordination. We will look at how the brain, specifically the hypothalamus and pituitary gland, integrates these two systems. You will need to confidently compare the rapid, targeted electrical impulses of nerves with the slower, broadcast chemical hormones circulating in your blood. Understanding these differences helps explain how the body matches each control situation to the right communication system.
Finally, we turn our attention to plants, which lack a nervous system entirely. Despite having no nerves, plants exhibit highly coordinated responses to environmental cues using precise chemical messengers called plant growth regulators. You will investigate how auxin drives directional growth in phototropism, how gibberellin triggers amylase synthesis to break seed dormancy, and how abscisic acid acts as a stress hormone to force stomata closed during a drought.
By the end of this unit, you will be able to explain the exact ion movements that generate an action potential, sequence the precise events at a cholinergic synapse, detail the sliding filament model, and map the molecular pathways of plant hormones. This unit is heavily tested in Paper 4, so mastering these rigid chronological steps and using precise molecular terminology is your key to the top grades.